Hello, dear students. Today we will discuss the supplementary factor 9:3:4 ratio in the series of gene-gene interactions which we are learning. In this type of interaction, which is also called recessive epistasis, the two independent dominant genes they interact with each other to produce a phenotypic expression which is different from what the single gene produces alone. Means a single gene has its own expression, but when it interacts with other gene or other supplementary gene, it forms a different type of phenotype due to the gene-gene interaction. Now, for an example, in sorghum or jowar, capital P means dominant gene shows blackish purple color of grain, whereas the small p shows the recessive color which is brown. Now there is a modifier factor Q, dominant Q, which is born on the other chromosome and alone it has no expression. But when it interacts with capital P means the dominant character of grain coloration, it modifies its action and now produces a reddish purple grain instead of a blackish purple grain. The color of grain changes due to the interaction of P and Q. The recessive allele of capital Q means the small q has no effect on the action of either P dominant or P recessive. Now this example can be explained by solving as follows. If we breed a blackish purple variety, blackish purple here, the black color is coded by these two alleles, dominant one, with a brown variety where small p is denoting the brown color. Now form the gametes P with Q and small p with capital Q. The F1 generation on fertilization is all reddish purple. As you can see here, capital P is present and Q has no expression as we have told. Still, instead of blackish purple coloration, the progeny is all reddish purple because Q has modified the expression of P. On selfing of F1 generation, which we got red purple, reddish purple and reddish purple, now we are selfing this F1 progenies, form the gametes. The gamete formation will be P with Q, PQ, P with small q, this gamete, small p with Q and small p with small q respectively. Now arrange these gametes as male and female on a checkerboard and make pairs vertical and horizontally. In this way, we will get 16 individuals in this checkerboard. Now we can observe that this capital P and capital P, which is dominant for blackish purple color, is associated with the modifying factor which is dominant Q and Q. This will now produce reddish purple color. Similarly here reddish purple, reddish purple, reddish purple, reddish purple, this will reddish purple, reddish purple, reddish purple and reddish purple. Now on the other hand, we can observe that the individuals having the genotype capital P and small q here, they will express blackish purple color. They will be black because the Q is not in its dominant form. This Q, which is a modifier, is actually recessive here. Hence, the action of the expression of P is not changed and it will produce its blackish purple coloration. Now, in these four individuals where P is in its recessive form, it will produce brown colored grain because Q modifier has no effect on this recessive gene. This has no effect on this, no effect on P and no effect on P. Therefore, for these 4 individuals will be brown. Hence, at the end, we will get 9 reddish purple, 3 blackish purple and 4 brown. Now, the explanation is the gene P and Q are the supplementary genes which are changing the expression of each other in this type of cross. A similar example is from the common house mouse, which have 3 types of coat colors. As we know, these are having 3 types of coat colors: black, albino and agouti. This black is coded by capital C, means the dominant gene, albino by another dominant gene A, and agouti, when these two supplementary genes interact with each other, they produce agouti colored generation. If we breed a true black breeding mouse with a true line albino, black because of the C capital and albino because of the A capital, the F1 generation will be agouti, neither black nor albino, because this is due to the supplementary action. The C and A, both dominant genes, have interacted and a third type of color is formed. Now form the gametes and form the F2 generation like the same we have done. In this way, wherever these both dominant genes for C and A are present, either in homozygous or heterozygous form, they will all be agouti, means all 9 will be agouti. The rest 3, where A for albino is recessive, will be black, and those 4 individuals where C for black is recessive will be albino. Here gene A and gene C are supplementary, hence they are producing 9:3:4 effect. Thanks.